Hello, I'm Matt Peterson, the CEO of TheraTarget. Thank you for taking time to preview TheraTarget's optimized immunotherapy assets. We'd like to introduce you today to our patented immuno-oncology landscape-altering, potentially market-resetting innovative technology. We'd also like to share with you the accessible market and our fully engaged team. As you're aware, over the last 10 years, cancer immunotherapy has advanced but not yet reached its full potential. TheraTarget's immunotherapy assets address a number of optimization goals.
TheraTarget's biodegradable dye-block HPMA platform delivery of the anthracycline epirubicin, KT1, induces immunogenic cell death and primes tumors for later multivalent synthetic peptide PD-L1 antagonist immune checkpoint interactions with MPPA. KT1 is first administered intravenously and provides a long circulating time with minimal toxicity. Solid tumors show enhanced permeability and retention caused by abnormal vessel architecture and reduced lymphatic drainage. These factors cause extravasation and preferential tumor accumulation. Combined intratumoral injection enhances efficacy and further reduces toxicity. The KT1 is endocytosed by the tumor cell and avoids the P-glycoprotein drug efflux pump. The endosome then migrates through the cytoplasm and merges with the lysosome. The lysosome's catalytic enzymes free the epirubicin, which migrates into the nucleus. In the nucleus, the epirubicin then intercalates into the DNA, causing inhibition of topoisomerase II and DNA replication, as well as inhibiting RNA and protein synthesis, resulting in toxic free radical production with lipid peroxidation, ultimately culminating in immunogenic cell death. KT1's induction of immunogenic cell death in heterogeneous tumor cells improves the immune response to the tumor. Antigen-presenting cells display the diverse tumor antigens to CD4 and CD8 positive T cells, which activates an array of T cell responses through lineages of activated T helper and cytotoxic T effector cells. The activated T helper and cytotoxic T effector cells recognize tumor cells and elicit a robust immunologic response with tumor cell destruction. However, some tumor cells present surface PD-L1 ligands that combine with PD-1 on T cells, causing deactivation of T cells. Over time, more and more tumor cells with PD-L1 ligands avoid immune surveillance, causing continued tumor cell growth and metastasis. To optimize tumor immunotherapy, HPMA delivers the multivalent synthetic peptide PD-L1 antagonist MPPA, which causes checkpoint inhibition. In addition to PD-L1, PD-1 checkpoint inhibition, the MPPA platform's cross-linking of multiple PD-L1 ligands on the cell surface causes endocytosis. The endosome then migrates and merges with the lysosome, where catalytic enzymes destroy the PD-L1 ligands. The combination of IV as well as local dosing of KT1 and MPPA results in the following advantages: significantly reduced adverse effects, robust targeted immunogenic cell death, a diversity of antigen presentations, multivalent checkpoint inhibition, first-in-class PD-L1 ligands recycling blockade, immunosurveillance capabilities, local and distant tumor responses, a special applicability for breast and lung cancers, and time for modulations of potential adoptive cell therapies. TheraTarget's durable immunotherapy with KT1 and MPPA has shown dramatic efficacy in multiple solid tumor preclinical models. TheraTarget's optimized immunotherapy addresses a critical need for new therapies in solid tumors that have combined global markets of nearly $100 billion by 2026.
Dr. Jindrich Kopecek, the world-renowned HPMA polymer chemist, designed the dye-block HPMA copolymer, which can attach therapeutic agents such as the FDA-approved topoisomerase 2 inhibiting immunoactivating epirubicin, which is KT1, and the trifold first-in-class multivalent polymer peptide PDL1 antagonist, MPPA. The dye-block copolymer carrier platform provides long circulation times and advantageous subcellular trafficking to improve tumor uptake, as well as reduce nonspecific side effects in solid tumors. The National Cancer Institute's Nanotechnology Characterization Laboratory has already characterized KT1 and has started their independent characterization of MPPA this year. As you're aware, this includes some of the IND-enabling studies. To further clarify the unique characteristics of KT1, the left panel depicts its ability to convert immunologically cold tumors to hot tumors by causing immunogenic cell death and releasing a robust portfolio of antigens. Furthermore, it reduces the myeloid-derived stem cells and T regulatory cells and recruits CD8 and natural killer cells. These utilitarian immunoenhancing activities have broad applicability in optimizing immunotherapy for multiple indications. The multivalent peptide PDL1 antagonist MPPA causes PDL1 checkpoint inhibition, but additionally reduces the pro-oncogenic activities noted in Section A of the right panel by reducing the recycling of PDL1, which occurs with monoclonal antibody therapy. This cellular activity occurs through the multivalent nature of MPPA, which by receptor cross-linking induces endocytosis of the cross-linked PDL1 ligands and destruction within the lysosome, shown in Section C. Without overwhelming you with data sets, I have summarized key preclinical findings in tumor models of breast, colon, lung, ovarian, and pancreatic cancers. The details are found in slides 13 and 14 for your review at a later date. As noted, the key features of KT1 on the left are its immunogenic cell death and immunoactivation, with increased overall survival rates in breast, colon, lung, ovarian, and pancreatic tumor models. MPPA on the right causes PD1, PDL1 immune checkpoint inhibition, reducing PDL1 recycling and immunity-enhancing tumor microenvironment reformation. Within the marketplace, both KT1 and MPPA have manufacturing, stability, and administrative advantages, with markets of over $20 billion and $117 billion, respectively. Inflection points in the commercialization pathway offer a full array of exits, including out-licensing, spin-offs, mergers, as well as acquisitions. KT1 and MPPA agents have no structural or functional equal, and with their stealth-like properties, adverse effects associated with each are expected to be less than the current therapies of pegylated liposomal doxorubicin and monoclonal antibodies against the immune checkpoint. With the National Cancer Institute's Nanotechnology Characterization Laboratory characterization of MPPA due late this year or early in 2023, we are poised, meaning that the fuse is lit and the protocols are prepared to proceed with, number one, pre-IND and IND-enabling studies for KT1 and MPPA estimated at approximately $3 to $4 million, and 2, a phase 1A, 1B on KT1 estimated at about $12 million, with the potential for out-licensing, spin-off, merger, or acquisition of the KT1 asset. The MPPA clinical trial will follow, as we anticipate it will be largely funded by the exit action chosen regarding KT1. The KT1 phase 1A, 1B will focus on immunologically cold refractory ovarian cancer. I want to emphasize that the principles and milestones discussed are not only pertinent to platinum-resistant refractory or recurrent ovarian cancer, but also to refractory breast, colon, non-small cell lung, and pancreatic cancers. Thus, the HPMA platform and these agents have multiple clinical networking nodes that will enhance their clinical application. As you're aware, this platinum-resistant refractory or recurrent ovarian cancer has a dismal prognosis and an unsettled later-line treatment clarity. Traditional non-platinum monotherapy avoids objective responses in less than 15% and a progression-free survival of only approximately 4 months and a life expectancy of approximately 12 months. PARP and VEGF inhibitors, along with other novel interventions, are changing the timeline, but unfortunately, a substantial proportion of patients eventually find themselves in this refractory category with unmet therapeutic needs. Recently, the Javelin 200 study identified platinum-resistant refractory or recurrent ovarian cancer patients who expressed PD-L1 and CD8 as a potentially responsive subset for combination chemoimmunotherapy. The progression-free survival hazard ratio of 0.53 and overall survival hazard ratio of 0.53 were promising. However, this treatment comes with significant impacts on quality of life, monitoring, and costs. This set of circumstances is the ideal opportunity for an orphan drug application for a phase 1A, 1B entry of KT1, as well as for MPPA. The accessible markets for KT1 and MPPA demonstrate great potential for multiple indications. The left panel demonstrates the KT1 market opportunity with 1, 5, and 10% penetration levels. On the right, the MPP market opportunity currently has a cap of estimated at $117 billion in 2026. This graphic also demonstrates the 1, 5, and 10% market penetration. I'd like to introduce to you the TheraTarget team. To collectively characterize us, I would say that we are driven, dedicated, and collegial. All of us have senior management experience over multi-million-dollar programs and are committed to the commercialization and success of this endeavor. Briefly, Jindrich Kopecek is world-renowned, fully engaged, and has a comprehensive background and knowledge of the technology and their design. David Nowotnick has years of relevant experience with polymers and successful clinical product launches. Jeff Harps has business acumen, management credentials, and experience with startups. Dr. Gandhari has perspective, experience, and connections to assist us in our commercialization. I bring to TheraTarget clinical trial experience in all phases, successful development and launch of medical devices, and have also practiced in Australia, the location where we feel we will conduct the clinical trial. TheraTarget is seeking funding for IND-enabling studies and the subsequent clinical phase 1A, 1B trial of KT1. After concluding the KT1 trial, An out-license, spin-off, merger, or acquisition are potential options which will supply funding for the MPPA clinical phase 1A, 1B. The deliverables are substantial, with a utilitarian delivery platform that increases circulation times, targets tumors, and reduces side effects. Both KT1 and MPPA can stand alone as well as in combination with each other or with other agents. There are multiple solid tumors with unmet needs addressed by TheraTarget's optimized immunotherapies. We have protocols, partners, which include Syndea and Paul Cronin, Curapath and Amador Garcia, and Still Meadow with Mark Holbrook. We are all prepared to move forward. Thank you so much for your time and attention, and we look forward to questions. Thank you.